Good day, this is Professor Will, CBMD PhD. Today is Thursday, May 14th, 2020. It is 3:21 p.m. Eastern Standard Time, and this is a continuation of my Corona series. So let's get started here. Tiny RNA that should attack coronavirus diminish with age. Disease. Date May 13th, 2020. Source Medical College of Georgia at Augusta University. Summary. A group of tiny RNA that should attack the virus causing COVID-19 when it tries to infect the body are diminished with age and chronic health problems. A disease that likely helps explain why older individuals and those with pre-existing medical conditions are vulnerable populations, investigators report. MicroRNAs play a big role in our body in controlling gene expression, and also are front line when viruses invade, latching onto and cutting the RNA, the genetic material of the virus. But with age and some chronic medical conditions, the attacking microRNA numbers dwindle, reducing our ability to respond to viruses. Much like not having enough troops on the ground in an actual war, the coronavirus is then better able to do what it does naturally, which is hijack our cell machinery so it can replicate. They have a longer-term goal of identifying the biggest hitters and replenishing those troops. They looked at the RNA sequence of actual two coronaviruses, SARS, which surfaced in 2002, and SARS-CoV-2, which causes COVID-19. And the sequence of the microRNAs that appeared to be attacking the virus, then used computer simulation to figure out which would logically fit together like puzzle pieces. Their pursuit included 4 samples of SARS and 29 samples of SARS-CoV-2, taken between January and April 2020 from 5 continents covering 17 countries, from the United States to Germany to Thailand. They found 848 microRNAs that target the SARS genome, and 873 microRNAs that target SARS-CoV-2 genome. They found 558 of the microRNAs fighting SARS also present in SARS-CoV-2, while 315 microRNAs were unique to SARS-CoV-2, and 290 were unique to SARS. MicroRNAs most proficient at attacking SARS-CoV-2 showed more than 10 target sites and might ultimately be found to be the most proficient at fighting the virus, which in a few months has changed much of the way the world functions. They also found the microRNAs targeting SARS-CoV-2 were associated with more than 72 biological processes, from the production of molecules to the immune response, and that many are known to become dysregulated and or diminish in number with age and with underlying medical conditions like diabetes and cardiovascular disease, a likely factor in increased disease, presentation and death rates seen in these individuals, the investigators say. An example is microRNAs like MIR15B5P, which has a high affinity for SARS-CoV-2, but is downregulated in coronary artery disease. In healthy younger people, these microRNAs, whose nature is to bind to the virus, are more apt to do as they should and prevent replication. In the 29 worldwide samples of SARS-CoV-2, 19 had identical microRNAs, which indicates the virus has a fairly uniform presence internationally, and that any effective treatments or vaccines should have broad impact. Next steps include studies in culture and lab animals to ensure findings are consistent with the computer analysis of human microRNAs in this study. The most important and striking feature of COVID-19 is the increased case fatality rate in aged individuals. The investigators write with the CDC reporting that nearly half of the patients requiring hospitalization are age 65 and older, and these more senior individuals account for 80% of the deaths. From a research perspective, there is a key set microRNAs that are important in triggering this abnormal response and making older patients more susceptible. We are looking at microRNAs in general dropping, but there is a specific subset that is key. The question is whether we can target those as therapy. Cocktails of multiple key microRNA, potentially given through the nose, might help restore sufficient levels of the key virus fighters, the investigators say. They already are moving toward producing synthetic microRNA that could supplement this frontline weakened by age or disease. Future studies also include pinning down which microRNA would be most impactful as an adjunct therapy, for example, with the drug remdesivir under study now for COVID-19, which works to stop the virus pirating of healthy cell machinery. Another question to pursue is whether some younger people who also are seriously sickened by SARS-CoV-2 infection already don't make sufficient numbers of some of the key protective microRNA. The microRNA present in the cells of our body typically target both 3' UTR, 3' untranslated region of the virus, the section of messenger RNA that contains regulatory regions that influence gene expression and protein function, as well as the coding region that ultimately produces a protein unless they are outnumbered. Normally your immune cells would go in and destroy them, but you have this large viral load as they continue to replicate and have all this abrupt inflammatory response, which ultimately results in the cytokine storms that help destroy rather than protect organs. Researchers think the reduced number of the key microRNA critical to the body attacking the virus is an enabler of the disaster then can follow. The genome sequence of SARS and SARS-CoV-2 were retrieved from the Gene Bank, the National Institutes of Health Genetic Sequence Database. The scientists used the whole viral genome sequence for microRNA target analysis. SARS, or severe acute respiratory syndrome, first surfaced in China and spread worldwide. But while it was more deadly than the current coronavirus, it was not as infectious, so less people ultimately died than are succumbing to COVID-19. Worldwide, 8,098 people were infected with SARS and 774 died, according to the Centers for Disease Control and Prevention. Near the end of the first week, in May, there were nearly 1.3 million SARS-CoV-2 cases confirmed in the United States alone, and more than 76,000 deaths. People aged 65 and older, and people of any age with underlying medical conditions are considered at higher risk for severe illness from COVID-19, according to the Centers for Disease Control and Prevention. Underlying medical conditions include problems like serious heart conditions, chronic lung disease, and moderate to severe asthma, and people with compromised immune systems such as individuals with cancer or who have had an organ transplant. Obesity, diabetes, chronic kidney disease, requiring dialysis, and liver disease are among the other conditions. Testing suggests 3% of NHS hospital staff may be unknowingly infected with coronavirus. Date May 12, 2020. Source: University of Cambridge. Summary: hospital staff may be carrying SARS-CoV-2, the coronavirus that caused COVID-19 disease, without realizing they are infected, according to a new study. Patients admitted to NH hospitals are now routinely screened for the SARS-CoV-2 virus and isolated if necessary. But NHS workers, including patient-facing staff on the front line such as doctors, nurses, and physiotherapists, are tested and excluded from work only if they develop symptoms of the illness. Many of them, however, may show no symptoms at all even if infected, as a new study published in the journal eLife demonstrates. The Cambridge team proactively swabbed and tested over 1,200 NHS staff at Addenbrooke's Hospital, Cambridge University Hospital, NHF Foundation Trust throughout April. The samples were analyzed using a technique called PCR to copy and read the genetic information of material present on the swab, producing a color change whenever the coronavirus was present in a specimen. At the same time, staff members were asked about the relevant coronavirus symptoms. Of the more than 1,000 staff members reporting fit for duty during the study period, 3% nevertheless tested positive for the coronavirus. On closer questioning, around 1 in 5 reported no symptoms. 2 in 5 had very mild symptoms that they had dismissed as inconsequential. And a further 2 in 5 reported COVID-19 symptoms that had stopped more than a week previously. To probe routes of possible transmission of the virus through the hospital and among staff, the researchers also looked at whether rates of infection were greater among staff working in red areas of the hospital, those areas caring for COVID-19 patients. Despite wearing appropriate personal protective equipment, PPE, red area staff were 3 times more likely to test positive than staff working in COVID-19 free green areas. It is not clear whether this genuinely reflects greater rates of transmission from patients to staff in red areas. Staff may have instead transmitted the virus to each other or acquired it at home. Staff working in the red areas were also swabbed earlier in the study, closer to when the lockdown was first initiated. So the higher rates of infection in this group might just be a symptom of higher rates of virus circulating in the community at the time. Nevertheless, extrapolating these results to the more than 500,000 patient-facing staff working across the NHS UK-wide suggests that as many as 15,000 workers may have been on duty and infected, with the potential to transmit the virus to co-workers, family members, and patients during the month of April. In fact, this figure could be even higher in settings where the supply of PPE has been very problematic. Test. and then test some more. Researchers explain all staff need to get tested regularly for COVID-19 regardless of whether they have any sort of symptoms. This will be vital to stop infection spreading within the hospital setting. Good day, this is Professor Will, CBMD PhD on infectious disease and microbiology. Well, hope you enjoyed listening. Until next time, have a great day.